Hey friends, welcome back to my channel Rajni Bio Academy. So today's topic is ABO blood group. So let's start. RBC plasma membrane contains antigen. So blood groups are classified based on the presence and absence of these antigens. There are more than 24 blood group system, but ABO is the most common one. Nature of ABO antigen: they are complex oligosaccharides present on glycoproteins and glycolipids. H antigen, which is a precursor molecule. So H antigen is like the foundation for A and B antigen, leads to formation of A and B antigen. H antigen is the base structure present in all individuals. They act as precursor for AB antigens. Structures include glucose, galactose, N-acetyl glucosamine and fucose. So different enzymes add specific sugars to H antigen to form A or B antigen. So let's talk about A antigen formation. So over here enzyme required is N-acetyl galactosamine transferase. So H antigen along with N-acetyl galactosamine in the presence of N-acetyl galactosamine transferase gives A antigen. So this reaction is very important. Next one, formation of B antigen. So over here enzyme required is galactose transferase. So H antigen along with gal transferase leads to formation of B antigen. O blood group, no functional enzyme required and H antigen remains unchanged. Genotype for blood group A, capital A, this is homozygous dominant condition, homozygous dominant, or we have one dominant allele and one recessive allele. Similarly over here homozygous dominant condition or one dominant allele or recessive allele. So this is the heterozygous condition. For ABO blood group we have dominant A and dominant B, and for O blood group we have both the recessive allele. So this is homozygous recessive condition. Next is blood groups and the antigens. So for A blood group we have A plus H antigen. For B we have B plus H antigen. AB A plus B plus H antigen, and for O only H antigen. No AB antigen present on O blood group. Dominance relationship. So IA and IB are codominant. These two exist in codominance, both dominant over I allele. So IA is dominant over recessive allele, IB is also dominant over recessive allele. Let's talk about antibodies in the plasma. So for blood group A we have anti-B antibody, for B we have anti-A, for AB blood group we have no antibodies, and for O we have anti-A and anti-B. Antibodies react with opposite antigen. For example, anti-A react with A antigen, anti-B react with B antigen, and mixing incompatible blood leads to agglutination. Very important. So here is the biochemical structure for A and B antigen. So this protein chain is constant. For A antigen and B antigen we have different compound attached to protein or lipid layer, and for this we have different lipid chain attached to different molecule. Basics of ABO blood group system: based on the presence or absence of antigen A and B on the RBC surface, plasma contain antibodies anti-A or anti-B. So this is the chart representation. If we have blood group A, then antigen on RBC will be A and antibody in plasma will be anti-B. For B blood group, B antigen and anti-A antibody in the plasma. For AB, A and B antigen and no antibodies. For O, no antigen and anti-A and anti-B antibody. Principle of blood transfusion: blood must be compatible between donor and recipient, that means same type. Incompatibility leads to agglutination or clumping plus hemolysis. Hemolysis means RBC destruction. Antigen antibody reaction: antibodies in recipient plasma react with donor RBC antigen and leads to hemolysis. Universal donor or recipient: so blood group O is the universal donor, no antigen present on O group, so not recognized by recipient antibody, so can donate to all blood group. Universal recipient: AB is the universal recipient, no antibodies present on them, can receive from all groups. Type A person receives type B blood group. Suppose if recipient is type A, the antigen is A and antibody is anti-B. Donor blood group is B type, so they have antigen B and antibody anti-A type. So over here what kind of reaction will take place? Anti-B antibodies bind to B antigen, donors RBCs, leads to agglutination plus hemolysis. Reaction in donor plasma: anti-A antibody bind to A antigen in the recipient RBC, agglutination plus hemolysis or clumping of RBC. So that's all in ABO blood group. See you in the next lecture.